bind to new proteins only after they've been synthesized? False. So, did I change this? No, okay, so that one is, oh.
It's false.
Yeah, is false, but I now see that I flipped the wording of how, so I asked which of the following is true. This one is, okay, this one's false right here.
You have one more question at the bottom.
Oh, okay, I was like, what happened? Is the next one none of the above? Okay, good. I hate it when I do that on exams, so that's why we proof each other, because I'll suddenly be like thinking of all these false things and then I'll forget that, anyway. We proof them very carefully. Okay, so and then so does it bind to new proteins only after they've been synthesized? No, it binds to them as they're being synthesized, just like HSP70 does. And BIP then acts as a winch, sort of folding this protein. And then I guess off the screen, or maybe it was an Instapoll, I have to go back and rework this slide.
It's in the handout.
It's in the handout. Excellent. Question here.
What happened is you were typing out like extra wording for this answer.
Oh, okay, and it pushed it down. Yeah, got too complicated. Okay, I'll fix it for the afternoon class. Question here.
If the option had said the BIP protein does not have an ERSS, would that be true?
The BIP protein does not have an ERSS. It would be ambiguous, and I think we originally had that and we modified it, and this is why. It would be ambiguous because the initial protein, as it's being synthesized, does have an ERSS. That's why it's soluble. And the reason you're wondering, is that true, is maybe you're recognizing that the mature protein lacks an ERSS, right? Because it's been cut off.
If you were to say mature BIP, then you're right.
Yeah. And these are called sometimes pre-pro proteins or something like that, the part that before it's been cut up. Other questions? Okay, excellent. Hopefully that served as a good review for that. And this is the movie that I showed you before. So all of these movies are, I mean, where I went up to a minute, all of these movies are on Canvas. They're in the module right there. They don't play in the PowerPoint that we've loaded there because the linking doesn't work once we upload them. But they're separate there in the movies, and there are more movies also to help explain some of these concepts. Another movie. Okay. So we've now covered how we have the N-terminus in the lumen of the ER, the C-terminus out. We've covered both of these. How then do we get a protein that spans the membrane multiple times? And in order to do that, once you understand how these two can be made, it's actually pretty simple to understand how you can have a multi-pass protein. And you now have all the tools to make any kind of protein you want, one that spans the membrane 7 times, 3 times, has its N and its C-terminus in any orientation. And the way that that's done, this is a nice picture from this is the equivalent, but I like this one from Cooper. It's just showing you that here's an N-terminus right here that's merged, and then we have a start transfer sequence, which they call an internal signal sequence. So that comes out and binds right here. This looks relatively short, so we know that the positive charges must be relatively here. And then something emerges, and if that were the whole story, this would just be an N and a C in the lumen. But look, we have a stop transfer sequence emerge, and that stops the transfer of the next segment through the translocon. And then we have here we have a second start transfer sequence right here that emerges. That would bind SRP, take it to the translocon again, and then start the transfer. And if this were the whole story, this would end up being a 3-pass protein, N-terminus on the cytosol, C-terminus on the inside that's emerging right here. BIP is, of course, binding all of these portions. HSP70 and other chaperones might be binding these portions. And there's other chaperones here as well. We've just focused on BIP. But this sort of shows that in progress we might have multiple start transfer sequences. So if we look at this figure from Alberts right here, we can see this is cool. There's a this is a 7-transmembrane domain. This is a G-protein coupled receptor. Here we have the hydropathy plot that we covered in another slide. This is in another lecture. This is the real hydropathy plot. You can tell because how jagged it is. And then the corresponding hydropathy plot with the protein with the signals are right here. And what I'd like, what would be a really good exercise for you to do is to first look at this and predict transmembrane domains. And then look at this right here. Black and white is sort of nice because they're not covered. And then try to go from here to here, and then go from here to here to try to draw the different patterns that you get in this complicated protein. And that will serve as a really great exercise for you. Questions? Okay. So now we're gonna move on now. We've covered all of the signals that are needed for integral membrane proteins to be made. We have all the tools to make any protein we want. We know all the ingredients that we listed before that were SRP receptor. We've added BIP as one that it helps actually in the translocation of a folding, because in folding the protein, it's actually helping the translocation. And those were all reconstituted by, again, the Global Lab at Rockefeller. And now we have the proteins in the ER, and now we have to look at how they are brought to other membrane compartments. So if the protein that we synthesized was a protein that was soluble in the ER, that would be something like collagen, fibronectin, insulin, growth factor, right here, it's going to end up on the outside world right here. And the path it's going to take is to go, so it starts its life by being soluble in the ER, and then it is packaged in a little vesicle right here that leaves the ER and fuses with the Golgi, where it receives other modifications, cis Golgi going up to trans Golgi, and then a secretory vesicle will take that protein and bring it up to the plasma membrane, fuse with the plasma membrane. So the membrane compartment of this secretory vesicle will fuse with the plasma membrane, releasing that soluble protein. So once a protein is soluble in the ER, it's never going to see the cytoplasm again, in most cases, if it's folded properly as a proper protein. Instead, it will be in the lumen of the ER, then in a lumen of a vesicle. When that vesicle fuses with the Golgi, it's going to be in the lumen of the Golgi. And then as it leaves the Golgi, it's going to be encased in a vesicle that buds off from the Golgi. Again, not seeing the cytoplasm, and then that vesicle will fuse with the plasma membrane. Similarly, anything that's if this were a transmembrane protein, and I guess we don't have that drawn right here, but if you picture a transmembrane protein right here in the lumen of the ER, as you picture it in the vesicles, what was in the lumen of the ER of that transmembrane protein, so in this case, if this were just taking one of these, if this were the N-terminus, that's in the lumen of the ER, that's never going to see the cytosol. Just the C-terminus that was there right here will be in the cytosol. And as it forms vesicles and whatnot, the C-terminus will always be in the cytosol. When this transmembrane domain, when this integral membrane protein is in a vesicle and fuses with the plasma membrane, what was in the cytosol, that C-terminus will be in the cytosol right here, and its N-terminus will be in the lumen, will be in the extracellular domain. So you can think of the whatever is put in the ER lumen as being destined for the outside of the cell, the extracellular portion of the cell, and what was in the cytosol, the cytosolic portions of transmembrane proteins, will end up being in the cytosol of the plasma membrane. Questions? That'll be more relevant as we talk about the modifications that occur in all of those steps.